The overall goal of this procedure is to monitor the process of transplasma membrane electron transport in real time through spectrophotometric assays.
This method can help answer key questions in the redox biology field regarding the mechanism of transplasma membrane electron transport and how a cell's redox environment is maintained.
The main advantage of this technique is that the assay is a real-time, multi-well, quick spectrophotometric assay.
To begin, seed C2C12 adherent cells at 2 × 10⁴ cells per square centimeter in rows A through F of a 96-well plate and cover the cells with growth media. Incubate the cells in a 37 degrees Celsius incubator with 5% carbon dioxide. After 24 hours in culture, aspirate the growth media, wash with 150 microliters of PBS, replace with differentiation medium, and incubate the cells for an additional 24 to 48 hours. Next, prepare stock WST1 and PMS solutions. Make a 10 millimolar stock solution of WST1 by dissolving 0.033 grams of WST1 in 5 milliliters of PBS. Then, vortex the solution. Then, prepare the stock solution of PMS by dissolving 0.0023 grams of PMS in 1.5 milliliters of deionized water.